The cell cycle. All life depends on cell division. It is the basis of reproduction for all organisms. This process replaces damaged cells and allows for growth amongst organisms. The cell cycle is a strategic set of events which starts when a cell is first formed via cell division to when it divides to create two cells. The cell cycle includes both growing and division phases. The growing stage, more formally known as the interphase, doubles everything from the cytoplasm to organelles to its chromosomal DNA. The division phase, or mitotic phase, is the actual divide or split of the cell. Seen in the drawing, most of the cell cycle consists of interphase. During this phase, the cell's metabolic activity is extremely high. The interphase is divided into three different subphases. The cell grows through each phase. The first is the G1 phase, the second is the synthesis phase, and the third is the G2 phase. During the synthesis phase, DNA is replicated, and during the G2 phase, the cell grows as it gets ready to divide itself. The mitotic phase consists of cytokinesis and mitosis. During mitosis, the nucleus, along with the duplicated chromosomes, divides and then are distributed to two daughter nuclei. Cytokinesis begins before mitosis ends and is the splitting of the cytoplasm. These two processes divide the cell into two daughter cells. Going a little bit deeper into interphase, during interphase, the preparation for division happens in three steps, as previously mentioned before. During the G1 phase, known as the first gap phase, organelles are copied and the cell grows physically larger. During the S phase, also known as the synthesis phase, the cell synthesizes a copy of the DNA in its nucleus. Centrosomes, microtubule organizing organelles, help separate DNA during the M phase. During the second gap phase, or the G2 phase, the cell continues to grow and make proteins and organelles. The cell also starts to reorganize its contents in preparation for mitosis.
After interphase, the cell cycle moves on to the step called prophase. This is the first of the 5 stages in the mitotic phase. During prophase, there are changes that occur in both the nucleus and the cytoplasm. Inside the nucleus, there are separate chromosomes formed by the tightening of the coils and folds of the chromatin fibers that are made up of a protein called histone combined with DNA. Each duplicated chromosome emerges as 2 identical sister chromatids, meaning they are pieces of identical DNA. The sister chromatids are joined together at the centromere. The mitotic spindle begins to form in the cytoplasm as microtubules grow out from the centrosomes, which begin to move away from each other. These mitotic spindles divide the chromosomes from a parental cell into 2 daughter cells. After prophase, the cell moves to the second stage of the mitotic phase, called prometaphase. In this phase, the double membrane structure that surrounds the nucleus, called the nuclear envelope, breaks into fragments and disappears. The microtubules that emerge from the centrosomes at the end of the mitotic spindle spread into the nuclear region and reach the chromosomes. Inside each sister chromatid is a protein structure called the kinetochore that is placed within the centromere at a specific DNA region. These kinetochores are attached to some of the spindle microtubules, and this causes chromosomes to become flustered and move. The spindle microtubules that didn't connect to the kinetochores make contact with microtubules coming from the opposite end of the centrosomes. Chromosomes are moved toward the center of the cell by forces exerted by protein motors connected with spindle microtubules.
During metaphase, the third stage of mitosis, the mitotic spindle is fully formed. The cell's chromosomes assemble on the metaphase plate, which is an area in between the spindle's 2 poles. The centromeres line up on this plate. The kinetochores of the sister chromatids then attach to microtubules from the opposite poles. The next phase, anaphase, occurs when the centromeres of each chromosome separate, splitting the sister chromatids. The kinetochores proteins then move the daughter chromosomes along the microtubules with the help of ATP until they reach opposite poles. Simultaneously, the spindle tubules that were attached to chromosomes shorten, while the ones not attached to chromosomes lengthen, moving the poles farther apart. By the end of anaphase, each end of the cell has the same number of chromosomes.
During telophase, the cell elongation that had started in anaphase continues. The daughter nuclei appear at the poles of the cell as nuclear envelopes form around the chromosomes. The chromatin fibers uncoil, and the mitotic spindle disappears. At the same time, cytokinesis occurs. Cytokinesis is the division of the cytoplasm equally between the two cells, and leads to the two daughter cells completely separating soon after the end of mitosis. In animal cells, such as the animal cell pictured here, a cleavage furrow forms and the cell pinches into two.